Hello everyone, welcome to iMeta paper interpretation. Today we'll introduce a recent study published in iMeta by Professor Wang Wei's team from China Agricultural University, titled Microbial DNA Methylation Links Rumen Transcriptional Activity to Methane Emission Divergence in Dairy Cows. Enteric methane is a key greenhouse gas from dairy farming, and it also contributes to 2% to 12% of feed energy loss. The rumen microbiome plays an essential role in feed fermentation and methane production. Most previous studies mainly focused on the composition and gene abundance of rumen microbes. However, even with similar microbial composition, individual dairy cows can show obvious differences in methane emissions. As a common epigenetic modification in prokaryotes, DNA methylation has been verified to regulate gene transcription and participate in microbial metabolism. Accordingly, this study puts forward a new research question: can microbial DNA methylation regulate ruminal methane production in dairy cows? This study has three major innovations. First, it establishes the first genome-wide DNA methylation atlas of dairy cow rumen microbes, providing a valuable dataset for rumen microbial epigenetic research. Second, although high and low methane emitters share highly similar microbial composition, their DNA methylation profiles differ significantly, which indicates epigenetic differences may contribute to varied methane emissions. Third, it connects DNA methylation, microbial transcriptional activity and methane emissions for the first time, offering an epigenetic perspective to understand rumen microbial function regulation. The results show that low methane-emitting cows have lower methane, carbon dioxide and hydrogen outputs, while the concentration of volatile fatty acids increases, especially propionate and valerate. Further analysis proves methane emissions are significantly negatively correlated with volatile fatty acid content. It suggests that metabolic flux may shift from methane generation to volatile fatty acid synthesis, so as to improve feed energy utilization efficiency. To explore the underlying mechanism, the research combined second-generation and long-read sequencing to build a high-quality rumen microbial reference gene catalog. The results indicate gene abundance is not fully consistent with gene transcription levels. It proves methane emission differences depend on not only microbial composition but also microbial functional status. Moreover, microbes in low-methane cows have more concentrated core metabolic functions, which means their rumen microbial ecosystems operate more efficiently. Afterwards, the research mapped the first genome-wide DNA methylation landscape of dairy cow rumen microbes. NMDS analysis shows methylation profiles can clearly separate in high and low methane emission groups. In total, 43 DNA methylation motifs were identified, and 36 of them have not been read in previous studies. These results indicate DNA methylation is closely associated with microbial transcription, and may participate in differences of cow methane emissions. The team carried out colocalization analysis of DNA methylation and differentially expressed genes to further explore the linkage between methylation and microbial functions. Among differentially expressed genes, the proportion colocalized with 6MA, 5MC and 4MC is 21.65%, 7.37% and 0.12% respectively, and 6MA shows the strongest correlation. For example, in Succinoclasticum, increased 6MA methylation is linked to higher expression of the propionate synthesis-related gene MMDA. This process utilizes more hydrogen, and reduces substrates for methanogens to produce methane, which suggesting that microbial DNA methylation regulates the transcription of key metabolic genes in methanogenesis, and further leads to methane emission differences among dairy cows. In summary, this study constructs the first genome-wide DNA methylation atlas of rumen microbes, suggests microbial epigenetic signatures related to high and low methane emission phenotypes, and correlates DNA methylation, transcriptional activity, and methane emissions. It extends rumen microbial research from traditional microbial composition and gene abundance analysis to epigenetic regulation, providing new insights to understand ruminant methane production mechanisms and achieve precise methane reduction. This is all for paper interpretation. Thank you for listening.